Imagine that you were able to slip inside a human cell and approach the spherical nucleus where the DNA is stored. The only access is through a ring-like structure, the nuclear pore. Passing through this strange gateway, you can see the double helix of DNA, which is carefully wound up on a series of tiny spools. These are the histones, and they are precisely shaped for storing the DNA molecule. Scientists were puzzled to find several tails projecting out from each spool. They wondered what might their purpose be, and it turns out that these tails have many functions. Some tails grab tight on the DNA, locking it in place. But when a section of DNA needs to be opened up, a tiny unlocking device, an acetyl tag, is attached to the tail by a special machine. Then the tail swings away, and the DNA is exposed and is ready to be used. This is just one tiny glimpse of an exciting new world of complexity. Beyond our DNA, welcome to the epigenome.
I find it fascinating how biology has changed in light of epigenetics. It used to be said that DNA rules our destiny. And in fact, many people would talk about the God gene that enables us to perceive of this being called God. Genes were sometimes said to be the culprits of many of these ills of society. The gene for obesity, the gene for this, the gene for that. And there may be some truth in this area, but Time magazine did a cover story on this topic, and they said their focus was going to be on why DNA is not your destiny.
Time's cover story was focused on the layer of digital information in the form of tiny switches, millions of which are attached to the tails of the DNA histone spools. Also, tens of thousands of tags are placed directly on the double helix. For example, 3 pink tags seen here, called methyls, bond directly to the DNA. They function as on-off switches, telling a gene when to be active and when to go to sleep. This discovery has presented medicine with a new key to improve human health.
Randy Jertle at Duke University is one of the world's leading experts on epigenetics. And he says that what makes him so excited about this area of study is that DNA no longer is your destiny. You can shape, you can change, you can remodel your destiny, because the epigenetic code, which sits on top of the DNA, comprised of millions of little tags and flags, can be remodeled, can be tweaked, can be literally reprogrammed for the next generation.
In 2009, the renowned cataract surgeon Dr. James P. Gills initiated a research project on the exploding field of epigenetics. After completing an initial manuscript, he invited Professor Tom Woodward of Trinity College to assist in research and writing. In 2012, Kriegel Publications released the mysterious epigenome, What Lies Beyond DNA.
What fascinates me about both DNA and the epigenetic code which sits on top of DNA is that we're learning so much more about the complexity of this programming. We are hearing scientists now talk about layer upon layer upon layer of very sophisticated tagging, flagging, and literally deploying of switches on top of the DNA. And I was really fascinated to study DNA and to write a book with Dr. Gills, Darwinism Under the Microscope. And so when he challenged me to join him in the project of writing a follow-on book on the mysterious epigenome, I was excited to learn about this area and was astounded at how powerful this revolution, the epigenetics revolution, has become. I even asked him his point of view on his writing of the book as well.
In a single cell, it was more complicated than a single city working. And the magnitude of that complexity made me very interested in writing that first book with you, Darwin Under a Microscope. And then when the concepts of the epigenome came out, and when I started reading about them, almost 2 decades ago, they started writing about it. In a decade that you'd see really publications coming out that were small, but they were definitely there. Now, epigenome is anything that is other than a genome itself. Can be splicing, if you use the term liberally. It can be anything that can affect the genome's metabolism and the dance of that genome. Because that gene isn't just standing there in a double helix. It's dancing and it's dancing to its conductor. The conductor is the epigenome. and it conducts it into different types of tissues. I got very excited about this because this showed not only that there was a depth to the biology, a greater depth than anybody wanted to recognize, particularly a Darwinist, but it was a depth to this that made the concept of a God creator, if you want to call it intelligent design, really a very valid stand.
In The Mysterious Epigenome, Drs. Gills and Woodward describe the sheer elegance and complexity of these new codes and signaling systems, which are often described collectively as the director of the DNA orchestra. Besides the tiny letters that decorate the histone, there are much larger chemical tags that shuttle back and forth among the histones, playing a vital role. Yet the most practical lesson and the focus of this research is how we can reshape our own epigenetic code within our body, and how we should alter our lifestyle to help build a healthier life for ourselves and for the next generation.
We can temporarily control our epigenome by the way we think, the way we exercise, the way we eat. Mutation is different. That's not reversible. Epigenome is reversible, but a mutation is done once and you don't bring it back. And usually it's not for the good. But an epigenome we can bring back. We can even take some of the bad epigenomic things that are going on in our body and change them with nutrition, with exercise, and with the way we think.
The epigenome is a marvel of integrated design, with its layer upon layer of coded information comprised of chemical tags and flags. This new world of epigenetics is very difficult to account for by the blind, undirected forces of nature, as Darwinism claims. In addition, evolutionary biologists have suffered the collapse of one of their favorite supporting arguments, the idea of junk DNA. The evidence against this doctrine has become compelling in recent years, as reported by biologist Jonathan Wells, author of The Myth of Junk DNA.
You know, scientists used to talk about DNA as a great big library, sort of a digital hard drive encased within the cell. And about 3 billion letters are arrayed across that disk, if you will, the hard disk of our cells. But only about 2% of those codes, those genes containing information for proteins, were seen as active. The rest was assumed to be broken refuse, basically junk that had been creeping into our genetic hard drive over eons of evolution. Well, that view is now under heavy assault from the best scientific evidence that we have coming in from labs around the world. In fact, many scientists have now said that the junk DNA concept is being replaced by the hyper-functional DNA view, that about all, 90-95% of our DNA is functioning and is doing a myriad of tasks which scientists even didn't know about 10 years ago, many of these jobs or functions. What's interesting, though, is that the junk concept does come into play at the epigenetic level, where we can turn good epigenetic programming and make it junky by, for example, bad life habits. Some children in northern Sweden were found to have had their epigenetic programming damaged because they were binging, especially the boys, during bumper crop years, and their children and grandchildren suffered a loss in their lifetime, expected lifetime of 30-some years. That's what we call damage, or maybe in this case, junky epigenetics. But those changes that have been before ill can be reversed, they can be changed for good in the next generation.
When we look at a single cell, we see a lot of particles. We see 1 trillion particles within a single cell. All these are integrated together in a functional manner. It's very complex, and people call it irreducibly complex because all of them have to work together and they each play off of each other for life. And all of these multitude of factors must work together. Therefore, for every one of them to work, they all must work. And that's why it's irreducibly complex.
In light of what we are learning about epigenetics, should we now be asking not how did all of this evolve by random accidental acts of nature, but rather, who created this biological wonder within ourselves, and what else can we learn about His great design?